Hello everyone, welcome back to Biophilia, your all-time study mate. Today we are going to see about the part 2 of the chapter that is about nephron. So if you like this video and get some help, then don't forget to like, share and subscribe to our amazing channel Biophilia. So here in the part 1 of the chapter we had seen about the parts of human excretory system. So हमने देखा था about the kidneys, then through the kidneys we went to the study of ureters. From ureters we had seen about a bag which stores urine, that is the urinary bladder, and then from the urinary bladder to the external environment is the urethra. So what is going to be nephron related to these? So kidneys जो हमने देखी थी, that we had seen the external appearance, how does it look, what is the color, the size, the weight, etc. But internally what is going to constitute the kidney is nothing but the nephron. So nephrons are also called as uriniferous tubules. Why? Because these are the tubules which are going to form urine. So for the formation of urine, or since they are related to urine, it is called as uriniferous tubules. So these are nothing but the structural and the functional unit of kidneys. So you might have heard about that structural and functional unit of body is going to be cells. So similarly, the structural and functional unit of kidney is going to be the uriniferous tubules or the nephrons. So each nephron is going to have two parts. First part hota hai glomerulus, second hota hai the renal tubules. So now what is glomerulus? So here I have tried to color code with the diagram present adjacent. So glomerulus is going to be a tuft of capillaries. So what is it exactly? We are going to see in detail in a few minutes. But just for now, understanding glomerulus, you can try to locate it in the diagram as per the colors. And then the second part of the nephron is the renal tubule. So renal tubule is nothing but different sized tubes which are going to come together and form the renal tubule. So different locations पे there are going to be different names. So first is the nearmost to the glomerulus is going to be the Bowman's capsule. So again check the colors and check the labelings in the diagram. You will get a benefit of it. So next is the proximal convoluted tubule, the one in the blue, that is the PCT. Then the green colored is going to be the loop of Henle, and the pink colored or the purple colored one is going to be the distal convoluted tubule, that is the DCT. And finally the last part of the renal tubule, that is the collecting ducts. So in this video we are going to see in detail about all the parts of the nephron. So we are covered with the structural and functional unit of kidneys. So let's get ahead. So here we see the first part, that is glomerulus. So this is going to be a tuft of capillaries. Tuft of capillaries यानी ये जो blood vessels आते हैं. So the one that is called as the efferent arteriole. Efferent arteriole जो होगी, it is going to come and it is going to divide into various amounts of small capillaries. पूरा गुच्छा बनाएगी. What is tuft? Tuft is nothing but गुच्छा, एक ऐसा बहुत ज्यादा जाली जैसा form होगा, and that is going to be the tuft of capillaries forming the glomerulus, and that is coming from where? It is coming from the efferent arteriole. So efferent arterioles break up hoga to form into tuft of capillaries, and this is going to be forming the tuft, that is glomerulus. अब ये tuft जैसे ही form हो जाएगा, finally these are also going to come and go together into, and these will again join and form the efferent arteriole. So ये efferent हो गया, and the one which is at the starting is going to be the efferent. So how can you remember whether efferent is coming or efferent is coming? So you can remember it in the way that efferent is for exit, E is for efferent, E is for exit. So that is leaving. So efferent coming from the renal artery is going to come and form the glomerulus, and finally the tuft of capillaries is going to come and join to form the efferent arteriole. So here also in the picture you can see about how there is going to be formation of the efferent arteriole into tuft of capillaries and then joining together again to form the efferent arteriole. So efferent, E is for exit. So coming the blood from the efferent arteriole will come into the tuft of capillaries, and then it will come out of the efferent arteriole. So this is the pathway for the blood. So अब ये जब capillaries में आ रहा है, what is going to exactly happen? So once it comes into the capillaries, there is going to be a renal tubule which is surrounding it. So that space that is surrounding is called as glomerular capsular space. So उसके पास में क्या होने वाला है? There is going to be a renal tubule which is called as the Bowman's capsule. So Bowman's capsule is going to be blind. That means it has only one opening. So कौन सा opening है? The opening which is going to be leading to the PCT, that is the proximal convoluted tubule. So ये side से the part which is nearer to the glomerulus, it is going to be closed, that is it is blind, and there is going to be doubled walled cup shaped structure. So Bowman's capsule, so you can imagine this is your Bowman's capsule, and in the Bowman's capsule over here you have your tuft of glomerulus, that is tuft of capillaries, glomerulus. So this is your Bowman's capsule in red, the yellow one is your glomerulus, and then the green one you can understand it as the PCT. So this is a little schematic diagram that you can understand for your simplicity. So Bowman's capsule में हमने देखा that it is double walled. So double walled, so we have to understand about what are the two walls. So first of all is the visceral layer or the inner wall. Visceral, that is the inner part. So visceral layer is going to be flat squamous epithelium and on the periphery, whereas there are going to be specialized podocytes. So podocytes, what do you mean by podocyte? Podo is related to the word that is foot. So foot-like cells which are going to have the podocytes in the remaining part. So these are going to have many interdigitate vegetations. So these are going to have some, finger-like structures, foot-like structures coming together, and they will be intermingling together to form an interdigited place, which is nothing but the feet. So it looks like feet, so it is called as podocytes. And what are these going to help in? These are going to help to form the slit pores or the filtration slits. So जैसे छन्नी होती है या sieve होता है. So similarly, as you have a sieve to separate out, जब चाय बनाते हैं तो चाय में से चाय पत्ती को हटाने के लिए जो आप mesh यूज करते हो या sieve यूज करते हो, similarly the podocytes are going to be helping in the Bowman's capsule, of course. So it is going to act like a sieve so that the urea and the other waste products can be filtered out from the blood, and the remaining whatever is needed is going out from the afferent arteriole. So what is the parietal layer? It is the outer wall, again made up of flat squamous epithelium, and here between the two layers, that is the visceral and the parietal layer, there is going to be some space. That space or lumen is called as capsular space. So here again you can see in the photograph as well. So the outer one is going to be your parietal layer, inner one being visceral, और बीच में जो जगह बच गई, that is the glomerular capsular space. So I hope you understood the first two parts, that is glomerulus and the Bowman's capsule. The next we see is the proximal convoluted tubule or the PCT. Now where is it present? It is going to be in the cortex. So as we have seen in the previous video between the kidneys, we had seen that kidneys are going to be having two parts, that is the cortex and the other one, that is the medulla. So if you do not remember this, then you can surely go and check out the part 1 of this chapter. Link will be provided down in the description box. So the part of the kidney, that is cortex, the PCTs will be present. And what are these? These are going to be highly coiled. So you can rewind the video and see the photograph of the diagram for the PCT. It is going to be very, coiled, and it is going to be surrounded by the peritubular blood capillaries. So how is it going to be from the point of view of cells? So cells, it is going to have the cuboidal epithelium with brush borders and long microvilli. Now why do you need microvilli? Microvilli because it has to absorb everything, because only the waste products should go out in the urine. अब चीजें जैसे ग्लूकोज हो गया या वाटर हो गया, these all are the substances that needs to be reabsorbed. These should not go in excess out in the urine. So these needs to be reabsorbed, and for the reabsorption the area should be increased. That is because of the brush border and the microvilli that is in the PCT. Now the cells are also going to have large number of mitochondria and even food reserves, so that the absorption and secretion process is not having any stoppage. Then comes the next part, that is loop of Henle. So here also I have tried to show you a photograph that will help you to understand that what parts we are speaking about. So this is your entire nephron. Nephron may we had already seen about the part that is the glomerulus. So the red one over here is the glomerulus. Then over here the cup-shaped blind double hole is your Bowman's capsule. Then the green colored part is your PCT over here. And then now we are speaking about the loop of Henle. So in the loop of Henle, it is looking like a hairpin look. Have you seen the hairpin? The hairpin is going to be like the U-shaped one. It is going to be the part which is going to descend down into the renal medulla. So PCT, where it was present in the cortex, whereas the loop of Henle, since it is going deep into the kidneys, it is reaching the renal medulla. So this is the striking point that you should understand. Loop of Henle reaches into the medulla, whereas PCT remains into the cortex area. So that is also very well defined. Cortex is the upper part, medulla is the lower deep part of the kidneys. Now the loop of Henle is going to be made up of two parallel limbs. So if you can very well notice in the part over here, the yellow colored one is going to be the loop of Henle. So in the loop of Henle, you see two parallel structures. So the one over here and one over here. These are the two parallel limbs. So one which is going downwards is going to be the descending limb, and one which is going upwards is called the ascending limb. So that makes up the entire loop of Henle. The next is the distal convoluted tubule. So after the ascending limb of loop of Henle is going to be continuing as the DCT. So we had seen about like this. This was your loop of Henle. This was your descending limb. This is your ascending limb. So after the ascending limb, you are going to see the DCT, that is the distal convoluted tubule. Now the distal convoluted tubule is going to just lie close to the Malpighian body, and it is going to be the coiled part of the nephron. So again you can see in the photograph here as well how coiled is going to be the DCTs. Now the collecting ducts. There are going to be many collecting ducts, very important to understand, and different areas we are going to open up into the renal pelvis, and from the renal pelvis they will be coming into the medulla and forming ducts of Bellini. So these are the things that you need to remember about the collecting ducts. So collecting ducts kya hai? Once it is forming the DCT, from the DCT they will be opening into what? They will be opening into the collecting ducts. So many collecting ducts will come together and open into the renal pelvis, because there are only, ऐसा नहीं है कि सिर्फ एक ही नेफ्रॉन है, many nephrons, that means many DCTs, and from many DCTs everywhere there is going to be one collecting duct. So एक हमारा nephron यहाँ पे हो गया. So here you can understand in a very beautiful manner. This is your nephron 1. Here you have a nephron 2, but between both, after the DCTs, they are joining to a common collecting duct. So यहाँ पे भी you can understand, यहाँ पे भी एक nephron होगा, यहाँ पे भी एक nephron connection होगा. Similarly, there are going to be many nephrons which will be opening into the collecting ducts. Next is the vasa recta. Vasa recta is going to be related to the efferent arteriole. So efferent arteriole, E is for exit. So now I hope you don't understand, like you don't get confused between what is it. So efferent, E for exit. So now you know what we are speaking about. So efferent arteriole, once it is emerging from the glomerulus, and it will be forming a fine capillary network. So पहले क्या हुआ? Renal artery आई, renal artery से there was formation of afferent arteriole. From the afferent arteriole, tuft of capillaries came up to form the glomerulus. Then they joined together to form the efferent arteriole. And now from the efferent arteriole, there is going to be formation of fine capillaries which around the renal tubule. So renal tubule में क्या क्या आता है? Renal tubule में you are going to have the Bowman's capsule, you are going to have PCT, then the loop of Henle and the DCT along with the collecting tubule. So all these are the renal tubules. So इनके पास में कौन होगा? इनके पास में होगा the different capillaries which will be running around, and these capillaries are nothing but called as peritubular capillaries. What are these called? These are called as peritubular capillaries, which are going to be coming from where? From the efferent arteriole. Now these are minute vessels. These are very small because these are capillaries. So capillaries, you know, these are almost one cell thick. So these are going to also form a U-shaped vasa recta. A U-shaped vasa recta is going to be formed, but it is going to be highly reduced or almost absent in the case of cortical nephrons. So now what are cortical nephrons? For this, you need to understand the next slide, that is going to be about the different types of nephrons. So types of nephrons, the one that is the cortical nephrons. So हमने अभी देखा था कि cortical nephrons में the vasa recta is almost compressed or almost absent in the case of these types of nephrons. So what are these? These are the majority of the types of nephrons. यानी जो majority mass of the kidney is going to be, then it is the cortical types of nephron. So in this case, what is the difference? The difference is on the base of loop of Henle. So loop of Henle is going to be very short and it will extend very little into the medulla. So as we had discussed previously कि दो part होते हैं, the upper one is the cortex, the lower one is going to be the medulla. So loop of Henle जो होता है वो generally बहुत deep जाता है, but in the case of cortical nephrons, these will just dip into the medulla and come over. So this is the type of cortical nephrons. The second type of nephrons that we see are going to be the juxtamedullary nephrons. So again, this is color coded for you to understand from the diagram. So the bright yellow one are the cortical nephrons which just touch about the medulla, and the juxtamedullary nephron, as in the name we have medullary, that means these are going to be going into the medulla, which are going to have very long loop of Henle. जैसे over here we have cortex and medulla. So in this case there are going to be very long loop of Henle, and because of that you are going to have the reach in the medulla at a very larger rate. So here you can have vasa rectas emerging out at a very large rate, क्योंकि ज़्यादातर जो vasa recta होंगे वो कहाँ पे होंगे? They will be near the loop of Henle. So because of loop of Henle is smaller in the case of cortical nephrons, the rate of formation of vasa recta is going to be lesser. So that is obvious thing that you should know. So juxtamedullary nephrons having large, longer loop of Henle going into the medulla, whereas cortical nephrons are going to have smaller loop of Henle just about reaching the medulla. So that is the two types of nephrons, and also we have completed understanding about all the different parts of nephron in detail. So here we are at the end of the understanding of nephrons. So that was all about for this chapter as well as for this video. And with this, if you like this chapter, if you got some help to understand it in a much more better way, then don't forget to like, share and subscribe to our amazing channel. We will be back soon with the next chapter that will help you to be following up with your marks.